So good afternoon. So you should see my presentation now. I would try to do in the next 20-something minutes to decomplexify a very complex situation. And I do not pretend to have all the answers, but if I can provide some beginning of answer, I think that would be a great move for everyone, and we will achieve that collectively. So here are my disclosures. So my research program on ME/CFS and on long COVID are supported by Open Medicine Foundation, and also more recently by the Patient-Led Research Fund for long COVID. And I'm serving on several boards as well. So where do we go from here? So we all be now well aware after over 2 years of pandemics that we are not out of the wood, if I may say, because we are to deal with these new situation that beyond the COVID pandemics, now we have to deal with the long COVID. And because we are, I think, well positioned, unfortunately, but that's true, that we know that probably over 50% of the long COVID will transiently become ME/CFS, and it's something that we probably think we can have a positive contributions to understand this very terrible situation, and the number are growing, and I think we have to react and eventually to learn from this situation. This is creating a very interesting window of opportunity, nevertheless, because we know the aggressor, so it's the SARS-CoV-2. We know the time frame, and we have the opportunity to study it on the very early stages of how is required to develop onset of long-term sequelae like ME/CFS, for instance. So there are challenges. So how can we recognize the multiple facets of long COVID? And I think this is a good example that we need to think outside the box to better understand long COVID and its consequences. So the best image I have in my mind is to try to solve the Rubik's Cube with changing colors. So I don't know if you are very good with the Rubik's Cubes, but I'm not. And if you change the rules and the color are changing from over time, that further complexifies. So as I told you at the beginning of my talk, I tried to decomplexify this situation, and now you know that I'm not very good with Rubik's Cube, so I don't know if I give you some confidence, but let me see by the end of this presentation. So our study population, we started with a study called SCOPIMED, which was funded by Open Medicine Foundation. SCOPIMED stands for Study of COVID-19 Long Haulers, Profiles for Detection of ME Development. And we put the focus on our research on non-hospitalized individuals during the acute COVID-19 phase, so which means that most of them were either asymptomatic or a very mild to moderate symptom. At that time that we selected patients in Quebec, all were infected with the Delta variant, so it's some kind of advantage. So we have a more homogeneous population, and all were naive to the vaccine because there was no vaccine available at that time. So we recruit patients with a minimum of 6 months post-COVID, and in average, they were around 9 months post their acute phase. But we have also access to the Quebec Biobank BQC, where we have access to hospitalized and non-hospitalized individuals during the acute phase and post this phase, as well as the equivalent in Canada that we call the CanCov Biobank, where we have also multiple samples that we can test at early time points if necessary. So what we'd like to learn here is, can we find methods and biomarkers that can allow us to identify and predict the clinical outcome, if we prefer the clinical trajectories of individuals suffering of long COVID, who will end up with ME/CFS or a related condition like fibromyalgia, who will be lucky enough to have a complete remission, and also what are the risks for other forms of sequelae like respiratory disease, other neurological disease that will be not ME/CFS. Also, how can we capture the sign of premature aging with other risk of developing more complex condition like diabetes, cancer, and other cardiovascular conditions that can be triggered by a long COVID. So this is what we are looking for. So SCOPIMED cohort is our discovery cohort, and we will do, and we are, thanks to the support of the patient-led research fund, will be in position right now to validate on a larger cohorts, not only from Canada, but outside Canada too. So we developed a very complex study design that we call a deep phenotyping that we combine with a provocation maneuver. And we are doing a longitudinal evaluation of our participants. So again, you see on this diagram that we start with a first visit where we use our stress test that we developed previously to study ME/CFS patients. And with these group, the stress test is generated by a passive exercise using an inflatable cuff that technically apply a therapeutic massage. But for ME patients is really inducing post-exertional malaise in 90 minutes of stimulation. So we have taking blood samples at a baseline and 90 minutes after induction of PEM. And at the same time, we will extract plasma and PBMCs. We are doing a neurocognitive evaluation with a mobile test that called Brain Check that in 15 minutes have a complete evaluation of different cognitive domains. We also, during the stress test, have a continuous monitoring of change in the brain oxygen levels, so that we call brain oximetry measurement using near-infrared spectroscopy, which are a non-invasive method with frontal electrodes applied on the left and right front. And also, we have also collecting first morning urine. We are also, during the stress test, use a connected vest called Exoskin, which allowed us to measure variation in the heart rhythm as well as respiratory rhythm. And we are collecting a saliva. Each participant must be able to complete self-report health questionnaire that you all know, like SF36, MEF20, DSQ2, and 1 week later after the stress test, the nurse are contacting the participant and they have to complete the DPMQ score, so which is to relate how severe were their PEM, and there's a way to score that, the PEM severity. We repeat the test simply at baseline plus 3 months later, as well as plus 6 months later, and finally, a year later, plus 12 months, we will repeat the first visit with, again, the stress test to see if we find additional change. So either the patient is improving over time or they are further regressing, which is sometimes, unfortunately, is the case. So this is a very long longitudinal study, and we do believe that using a standardized provocation maneuver is more likely to reveal changes, molecularly speaking or clinically speaking, and instead just to have more static measurement at different time points. So this is one major distinction in our study design compared to other studies. We start to search looking for ME/CFS signature among long COVID by studying the contribution of circulating microRNA. We developed a first diagnostic panels with ME/CFS that we published in 2020 in scientific reports. These 11 panels gave us a very high accuracy prediction of not only who is developing ME, but also to allow us to stratify very specific regulator that we call small microRNA, okay? And why we pay more attention to the small microRNA, so just to allow me to refresh your biology of genetics, is that we all know that we have a single database that is present in every single cell that we call the DNA. And this DNA is activated at different regions, so that will make some specific messenger that we call messenger RNA. And those messenger RNA are giving instructions to make muscle cells, brain cells, bone cells when they are activated in the right amount at the right time. And those messenger RNA are decoded by the ribosomal machinery inside the cells to make the functional products that we call protein. And as you may know, too little or too high or some modification in the protein leads to some diseases. So microRNA are very small non-coding RNA, and they are attaching normally at the end of the 3' end of the messenger RNA, and such attachment attract a complex of proteins that prevent the decoding of the messenger RNA. So the net outcome of such attachment will reduce the level of the targeted proteins. So we block protein production in very simple words. One microRNA can target up to 200 different genes, and one gene or one messenger RNA, if you prefer, may be targeted by several microRNA. So very complex, very powerful, small regulators. So why microRNA are interesting? Because they are very stable analyte in different biological fluids. We can use qPCR simply, so you can translate a diagnostic panel the next day in certified diagnostic lab if you wish, as well as we can use a panels that would give us a very precise and accurate diagnostic tools for a specific disease, or to stage a disease along different stage to predict either a prognosis or to predict a therapeutic response when necessary. So we published that in scientific report in 2020, over 24,000 downloads since November 2020. You can see on the diagram in the light blue the 11 microRNAs. So they have a nice name like MIR4433A5P, like it's a kind of postal code. And these microRNA are known to either predicted target or non-target in green, as you can see here. And what you can see in the light pink or yellow here are either pathways or biological situation where these genes are involved, like physical fatigue, like sleep disorders, like mitochondrial disorder, chronic fatigue syndrome, so function of muscles. So all of these microRNA are interacting with each other and are targeting very important targets that make a lot of sense in the context of myalgic encephalomyelitis. We use also the different profile after the stress test and before, so after 90 minutes of simulation and a baseline, to establish a variation in the expression level that allowed us to create initially 4 clusters. And now the cluster 2 is also further separated in cluster 2A and 2B associated with more severe specific symptoms. So we use these diagnostic tools to predict who will develop ME/CFS amongst long COVID sufferers, and why it's relevant is because when we, beside the fact that long COVID and ME/CFS have several overlap in terms of symptoms, including PAM. So that's why there is a great value to doing a provocation maneuvers to reveal PAM, or at least to induce it in a safely manner. But also you can see the very same microRNA here on this diagram where you can see they are connected with, of course, cognitive impairment, chronic inflammatory disorders, but they are connected also with the SARS-CoV-2 COVID-19. So there are key genes that are targeted by SARS-CoV-2 and also targeted by our 11 microRNA. So molecularly speaking, there are some interaction that sustain our view of using this 11 microRNA to predict the risk of developing ME/CFS among long COVID. So this is what we have done. So we combine the expression analysis of the 11 circulating microRNA with random forest analysis, which is a mathematical tools. And from that, the machine learning approach gave us a stratification of 6 subgroups. So one obviously is myalgic encephalomyelitis. Another group is fibromyalgia. Another group is ME/FM, because FM can be a comorbidity, and we published very recently another paper using the 11 panel to predict the risk of developing, not the risk, but the diagnostic of fibromyalgia or as it is or as a comorbidity of ME. But we also define three additional groups that are not ME related. One is respiratory diseases like COPD-like. The other one is other neurological disease, excluding ME/CFS. And the other one is, we call it for now, severe allergy, MCAS-like, but non-ME/CFS, but they are very close to be ME/CFS. We also further use, because we apply the stress tests in that context, we were able to further stratify long COVID developing ME/CFS with or without fibromyalgia along our 5 clusters, and you see the percentage distribution. And we more recently further subdivide fibromyalgia as a severe and moderate using cognitive assessment. So altogether, when you look about who is developing ME, fibromyalgia, or ME/FM in the long COVID cohort, it's about roughly 60% of the long COVID after 9 months post-COVID. So we are pretty sure that these individuals, unfortunately, are engaged in this long-term sequelae trajectory. And what we are doing right now is to further validate the 11 microRNA diagnostic panel using patients recruited at 6 months, 3 months, and even at the 1 month post-viral condition to further assess how robust is the panels to predict the long-term trajectories, as well as to further complement the actual microRNA for each of these subgroups, especially the new ones, to define additional panels to have a more precise diagnostic tools that will be in aid of clinicians that are dealing with those patients. We evaluate the cognitive domain. So this is a brain check report. You can see that we are measuring attention, mental flexibility, executive function, and memory. This is a very rapid test. In less than 15 minutes, patients are evaluated. We do the evaluation before and after the application of the stress test, and using a mathematical algorithm, we create 3 clusters that we call A, B, and C. And we look at different markers. So for instance, in cluster A, you see that we have a severe decline in the memory as well as in the cluster C, amongst others. The cluster C is the most impacted cognitively speaking. And this is interesting because the way that we can stratify, cognitively speaking, the long COVID participants, we can also further assess biomarkers that can reveal a contribution or a more precise contribution. And we discover in cluster C, for instance, a severe elevation of soluble SMPDL3B. And I will talk a bit more about that one because you will see it's a very intriguing but important protein. SMPDL3B may merge the brain metabolic and immunity disturbances that we are seeing in long COVID as well as in ME/CFS. So in this slide on the panel A, there is a diagram showing that SMPDL3B are expressed, is a membrane bound, normally protein on expressed at the surface of macrophage, but also expressed on dendritic cells, another type of immune cells. And they are also involved in the regulation of inflammatory process by inhibiting toll-like receptors. So very, very important actions. We also know, and this is a diagram generated by my research team using Ingenuity pathway analysis, that SMPDL3B play a key role in the conversion of sphingomyelins in ceramide. And we know that ceramides are a decrease in men and women suffering of ME/CFS, as well as sphingomyelin has been reported recently by the group of deriatronumas that were over expressed in the plasma of ME patient. So there is a connection between SMPDL3B and those metabolic changes, as well as you still see some connection with some of our microRNA that can interfere with those processes. What is very important, and that's further add to the complexity of long COVID and ME/CFS, is in the panel C, we can measure soluble, so the cleaved version of SMPDL3B. So when the protein is cleaved by what we call SHEDAs or some GPAs there is a free version floating in the blood, and we can measure that even on healthy individuals. And this is a normal situation. But in ME patient on long COVID, we find some cases where, let's say that there is an increased cleavage of membrane-bound SMPDL3B, which means that there will be almost no protein in the membrane, which means that that protein cannot play its role in immunity or in the lipid metabolism. And we will find a lot more soluble version in the bloodstream of long COVID or ME/CFS patient, which is a very abnormal situation. We have also a third case where still long COVID and ME patient, if you just look at the blood or the plasma, you will find almost levels similar to healthy individuals, which is not true, because in their case, they have a lack of capacity to produce enough SMPDL3B. And there are epigenetic mechanisms that can be involved, like DNA permethylation. So where you have CPG site in the promoter regions that are methylated, that preventing the transcription. So if you prefer the activation of the genes that are in charge of producing SMPDL3B. So we need to recognize these two situations because the treatment will be very, very different. In that case, we need to block the scissors, what is releasing SMPDL3B to have enough of the membrane. And in the other case, it's not by blocking the scissor, it's just by, we need to increase the level of production. So two different situations that needs precision medicines to move forward. So why SMPDL3B is important? So the sphingomyelin phosphodiesterase acid like 3B is involved with the soluble version, is associated with symptom severity in ME/CFS. In the panel A, and all these results will be published soon, but they are unpublished results. So this is from the Norwegian cohort of Dr. Flug. We have the same thing with the French-Canadian cohort. So you can see that there is a very steady elevations of the soluble concentration in the bloodstream, which is associated with the severity of the symptom. We also observe a change associated with age, and we know the reason for that, especially with females, because in the case of females, the decline of estrogen level around the menopause, around 50 years old, and that explains there's a direct correlation between the circulating level of the soluble version of SMPDL3B and estrogens, which also explains in panel C why females overexpress the soluble SMPDL3B when compared to male. And moreover, in panel D, we observe that when we stratify ME patient versus the ME suffering of POTS, there is a stronger elevation with the ME with POTS. And we don't understand fully the reason here, but that can give us some hints about how to treat maybe POTS in the ME or long COVID. Interestingly, when we further study our long COVID cohort, we saw the same thing. So you can see on the panel E and F that long COVID having over 23 nanogram per ml have a more severe PEM score. But interestingly, we have patients having very, very low level, below 15 nanogram per ml, which also have elevated score. So this is what we call in biochemistry a U-shaped response, which is quite common in some systems. And that if you have, again, the idea is if you have little membrane bound of protein, either you don't produce enough like here, or you have enough, but you cleave too much, you will have the same problems. We see that also for the cognitive score, which may explain what we observe initially that allowed us to chase SMPDL3B. We also, during our deep phenotyping, I told you that we are measuring or monitoring change in the brain oxygen level. So you can see on the panel A, B, and C a comparison of long COVID that is developing ME and ME patient, ME135, and a short COVID. The first observation is that the y-axis represents the brain oxygen level, which is never close to 100%. It's between 60 to 80% because it's a mix of venous blood, which is poor in oxygen, and arterial blood, which is rich in oxygen. And what you can see on the dark blue is the left side, and the brownish slope here is the right side. And you can see that there's a kind of left-right asymmetry in the oxygen level. So the oxygen decreased during the stress test, and going up. It's even more pronounced, the left-right asymmetry, which is typically abnormal. And you can see on short COVID or LT individual, both lines perfectly overlap. So it's a good sign that when we saw those abnormalities in terms of brain oxygen level, there's an immediate translation in change in the cognitive functions or impairments that we are measuring. So this is what we see in the panel D, where everything below the 15 percentiles that I put in red here are abnormal and significant change. So we saw that for the long COVID at baseline already, we have some issues in terms of mental flexibility and executive function. The Stroop test is not doing very well. And it's further exacerbated after the stress test. And we saw that this is a typical response for ME patient, that both immediate recognition at the memory level and delay recognition are impaired. This is a very typical response. And you can see on the short COVID, which fully recovered, there is no abnormality or at least significant abnormalities here. Moreover, here in the panel E, I report for you that we observe that when we have a very high circulating level of SMP-DLL3B, which correlate with less oxygen levels during the stress test, as opposed to individual having much higher oxygen level. Interestingly, we saw through the DSQ that, again, patients having high circulating SMP-DLL3B protein have more problems remembering things as opposed to patient having between 15 to 27 nanogram per ml. We apply different multiomic approach, and this is just an example that in panel A, we are able to stratify our long COVID in different subgroups, and we look at different markers. I won't go through each of them, but I would like to pinpoint your attention on FGF21, fibroblast growth factor 21. That factor was also identified in previous studies in cells in plasma of patients infected by SARS-CoV-2 compared to LT control with comorbidities. And you can see that FGF21 in this volcano plot was the one with the most significant fold change in terms of upregulation. And we studied that in the ME/CFS. So on the panel D, you can see the concentration on the y-axis of FGF21 in the plasma. This is comparing fibromyalgia group, ME group, ME/FM, and healthy control. And you can see that we have a significant difference between ME/FM and LT control. And when we stratify our cohort of long COVID in FM-like, ME-like, or ME/FM-like versus the short COVID, you see the same patterns that ME/FM are the ones that display the more significant gain in circulating FGF21 compared to their control, the short COVID. So same behaviors, which further sustain our view that first we can stratify patients and identify their clinical trajectories, as well as to using more conventional tools like biochemistry, we can identify relevant biomarkers. Why FGF21 is interest for ME and long COVID pathogenesis is because FGF21 in panel A is connected with cognitive or the cognitions, as well as the cardiac or cardiovascular function, more metabolic function center on the livers and the gut, as well as in the muscles. So all these muscles and tissues are affected in ME/CFS, as you know, and also in long COVID. So we saw that patients having extremely high FGF21, so over 200 picogram per ml, are not doing well in terms of general fatigue, panel B. In terms of autonomic score using the DSQ scoring system, also they are very significantly affected. You see even the sleep is more affected. And interestingly, mental fatigue, there is a nice positive correlation. And the most significant correction I would like to attract your attention here is the panel F, where the PAM or the DPAM-Q score after the application itself is highly significant positive correlation. So more is severe the PAM, and more you see the high level of circulating FGF21. The good news may be, and this is only a preliminary results, and I remind you, this is not a clinical trial, is we look at potential difference in the AMI group. So we have AMI that were not under treatment with low dose of naltrexone versus AMI receiving naltrexone. And even though that we still have so little individual in that group, you can see the trends, okay? It's not statistically significant yet, but you can see the big difference between those receiving low dose of naltrexone in terms of their plasma FGF21 levels. So I would say that when patients have a very high level of FGF21, I would be quite confident to recommend to the physicians to maybe apply a low dose of naltrexone here. So what are the key takeaways of my presentation? I think it's time to first work collectively and together to build a roadmap to shift from confusions to clarity. We learn a lot more now. And I think the use of a standardized provocation maneuver combined with deep phenotyping methods represent a very transformative approach that can accelerate our understanding of long COVID. The use of circulating microRNAs can be used to predict the clinical trajectories associated with long COVID, including the risk of developing ME/CFS, but also something else. And I just gave you two examples here that elevation of plasma level of SMPDL3B and FGF21 are associated with long COVID transitions to ME/CFS, and they represent potential therapeutic target. So finally, I would like to acknowledge a special thanks to all participants and my teams and collaborators and the private donor and also patient-led research funds and Open Medicine Foundation for their incredible and outstanding support towards my ME/CFS program and long COVID program. And a special thanks to several patient partners that are just represented here by the icons, but those patient partners are and were very instrumental in the development of our stress test. And without them, I wouldn't be here today to presenting you these exciting results. So thank you all for your time.
Thank you, Alain. So we are slipping behind the schedule, so maybe one question? Right at the back.
Yes, hello, thank you. I had one simple question. Did I understand it correctly that the patients had different oxygenation of the brain hemispheres? And
Yes.
how do you explain that phenomenon? And if by any chance, is it always one particular hemisphere that gets less oxygen? Or is it random?
We have tested over 200 ME/CFS patients and over 60 now long COVID. So there are different patterns and behaviors, okay? But more are pronounced the left-right asymmetry. This is one element that are really associated with cognitive disturbances. Beside the fact that often we saw patients that you see left and right decrease, so there are very significant decrease. To simplify the presentation today, I omit to tell you that we have also the information about the oxygenated hemoglobin and desoxygenated hemoglobin, so we can tell you if there is an increase in the cerebral blood flow. This is something that we are capturing during the stress test, or a decrease. And sometimes the increase and decrease are symmetrical, but sometimes they are asymmetrical. And they are sometimes so significant. So we saw in some patients, this is not the majority of the cases, but we saw change up to 30%. So everything above 5% is significant. So 30% is very dramatic, and they are often the most severe one affected cognitively speaking.
Okay,
Thank you.
I think we have to hold it there. So thank you again, Alain. Thank you very much for your presentation.
Thank you.
Thank you.